Hello again. Today we are going to talk about how the code is carried. In this lesson, we are going to define new concepts like transcription and translation. And we are going to understand that the genetic code is carried as a sequence of bases on the DNA molecule. So, the concepts that we are going to talk about today: transcription, translation. We have a new nucleic acid, which are mRNA and tRNA. Okay, let's start with the first point, which is how does the DNA in the genes instruct the cell to make particular proteins. So, we are going to learn how do we use DNA to make proteins. If you remember before, we said that DNA as gene controls the production of protein, which is responsible for characteristic. So, focus on this DNA. You will find that gene carries a series of coded instructions, which are code words to produce the proteins. So here in the picture we have 4 code words with 4 different proteins. And you have to know that each word code on DNA is made up of 3 bases, which are 3 letters. They are arranged in a certain sequence. So here, each 3 letters, they produce 1 amino acid. So then it turns into a protein inside the ribosome. And also we have another 3 letters, another amino acid, another and another. So we have 4 here, 4 word codes. Okay. So you have to know that each 3 letters or each word code is called triplet. So here we have the first triplet, the second, third, fourth triplet. Triplet means the 3 letters which produce the amino acid. And those triplets agree with a single amino acid. So each triplet agrees with a single amino acid in a protein. So the sequence of bases in DNA is a series of coded instructions for the building of amino acids into proteins. Okay, now I'm going to show you a little bit about the genetic code. So what I have here is the left half of a DNA molecule. And if I ask you to complete the right side of the DNA molecule, you should be able to fill in the bases that would bond in the complementary base pairing. So, C, G, T, C, C, A, T, T, C. If I ask you to complete this DNA code, you know that C connect with G and T connect with A, G with C, A with T. So, that's how you would complete. C's and G's go together, and A's and T's go together. That's pretty simple. Now, when you start learning about protein synthesis, we usually use part of the DNA molecule to act as a pattern for transcription and translation. Now, during the explanation of producing proteins, you will understand what we mean by transcription and translation. So, let's say that we are using this DNA. We have this DNA strand as the beginning of the transcription. So, this is half of my DNA molecule and would like to know what mRNA. So, mRNA is going to bond next to the DNA in the nucleus during transcription. So, DNA, half of DNA, will bond with another half to produce mRNA. So, this mRNA, okay, so I would have G for guanine, C for cytosine, A for adenine, G, U. Here, instead of using T, which is thymine, we use U, the fifth base, which is uracil. So when we make mRNA inside the nucleus, that will change the thymine base into uracil base. So instead of each T, that will connect with U. So T connect with A, but A will not connect with T, it will connect with U, which is uracil. So this is my messenger RNA. Messenger RNA comes into the nucleus and copies the DNA code. So here we have DNA inside the nucleus. mRNA comes to the nucleus and copies the DNA code. And the mRNA will take this code out of the nucleus and it will arrive to the ribosome. So again, we have DNA. mRNA comes to the nucleus just to copy the code of DNA. Then it takes the code out of the nucleus to go to the ribosome to produce the protein, or the nucleic acid, or the amino acid, to produce protein. Okay, so again, mRNA will take this code out of the nucleus and it will arrive at the ribosome. So now, let's say that messenger RNA has arrived at the ribosome. Now, messenger RNA here has arrived at the ribosome. Now, we have something called tRNA or transfer RNA. This transfer RNA is going to bond next to the messenger mRNA. So, instead of DNA connecting with mRNA, now mRNA will connect with what? With tRNA. So here we have the side of mRNA. This mRNA will connect with tRNA. So G with C, G, A, U. As we said, A will connect with U, not T, because out of the nucleus we don't have the base T. We only have U, uracil, instead of T. So again, C, G, U, C, A, U, C. This is the tRNA, which is transfer RNA. So here in the orange, this is my transfer RNA. And I might ask you, how many amino acids in this genetic code? What do you think? How many amino acids in this genetic code? As we said that each amino acid contains 3 bases. So C, G, and U, they will produce the first amino acid. C, and A will produce the second amino acid. U, and C, the third amino acid. So this would code for a particular amino acid, this one, particular amino acid, this code, and this code for another particular amino acid, and this the third code for another particular amino acid. So here we mentioned many concepts like transcription and translation. Now let's define transcription. So transcription is the first step in gene expression. Actually, it involves copying a gene's DNA sequence to make RNA molecule. So the type of RNA here is mRNA, which is messenger RNA. So making mRNA by using DNA sequence is called transcription. So transcription is making mRNA by using DNA sequence, as we explained before. But you have to know that RNA never contains the base thymine, which is T. Thymine is replaced by a fifth base called uracil. So thymine is replaced by a fifth base, which is called uracil, U. So instead of the base pair AT used in DNA, in transcription we have the base pair AU. Okay, all of that happened in transcription. Instead of AT, we will use AU because we are out of the nucleus. Now the second concept is translation. So translation is making a protein by using mRNA. As we said, mRNA code connect with tRNA and each three bases and each word code in the sequence will produce an amino acid. And all amino acids together that will produce protein. So the second concept, translation. Translation is making a protein by mRNA. But here we use tRNA. As we said, we use tRNA, which is a type of RNA molecule that helps decode a messenger RNA sequence into a protein. So tRNA helps decode a messenger RNA, which is mRNA, sequence into a protein. So the last thing, focus on all the process again. Here in the picture, we say that we have DNA inside the nucleus. Okay. 1/2 of the DNA strands connect with another strand to produce mRNA by transcription process. So transcription process, the base pairing rules G, C, and AU. Instead of AT in DNA, we have AU in mRNA. So GC and AU are used to make a messenger RNA, which is mRNA molecule, against one strand of the DNA double helix. So here we have two strands in DNA double helix. 1/2 will be used with mRNA to produce the messenger mRNA to go out of the nucleus. Then it reaches into the ribosome to produce protein by using tRNA. So mRNA with tRNA, they will produce the protein. Okay. Now, as we said here, tRNA with mRNA, they will produce many amino acids which produce the proteins. Now in your book, page 196, we have here how does the DNA in the genes instruct the cell to make particular proteins. As we said that each gene carries a series of coded instructions, code words, for the synthesis of proteins. Okay, and each code word on the DNA is made up of 3 bases. Look, here we have 3 bases in each amino acid, and each code word called triplet. This triplet corresponds to a single amino acid in a protein. So that's what we explained before. Now, passing the messages to the ribosomes. Here, this coded information in the genes is located on the chromosomes. You know that the chromosomes, they are inside the nucleus. Now, you have to know that ribosomes are protein manufacturing station. And those ribosomes are found outside the nucleus in the cytoplasm. So until now, you have to know that genes is inside the nucleus as chromosomes, and we have ribosomes in the cytoplasm. And ribosomes are the protein manufacturing station. Now the question here, how does the code pass from the nucleus to the ribosomes in the cytoplasm? We say that it's carried by another type of nucleic acid which called messenger RNA. So mRNA is one type of nucleic acid which can carry the genetic code to the ribosomes to produce proteins. Okay, this is the process. Again, mRNA moved to the protein with tRNA to the ribosome. They will produce protein. Okay, here in your book also we have transcription and translation. You have to know that the mRNA is made by a process called transcription. So we say that producing mRNA is called transcription. And you have to know that RNA never contains the base thymine. Thymine is replaced by a 5th base called uracil, U. So as we said, instead of AT in DNA, we use AU in transcription. Okay, here mRNA leaves the nucleus and travels to the ribosome. As we say, the mRNA leaves the nucleus and it travels to the ribosomes. Now you have to know that the sequence of bases in the mRNA is used to build up a sequence of amino acids into a protein in the ribosome. So mRNA with tRNA inside the ribosome, they will produce the protein. This process is called translation. Okay, here we have a summary of replication, transcription, and translation. Replication makes a DNA copy using DNA. So when we copy the DNA, we call it replication. Transcription making mRNA by using DNA code. So half went from DNA and half from mRNA. That's called transcription. Translation making protein by using mRNA. So here, mRNA with helping of tRNA that will produce proteins inside the ribosomes. This process is called translation. So this is the process again. Translation happened inside the ribosome, which changed the mRNA with helping of tRNA into protein. And here we have transcription using mRNA. So summary of the process: DNA to mRNA, then protein, and the protein responsible to give the characteristic in the body. And thank you for listening. Study well and please answer the questions page 197 and send them to me in B12. Good luck.